Okay, so for example, you can go and let's begin by clicking on amino acids. You see, it will not allow you to download, but it will open up for you. That's why I couldn't post them. We're not concerned with every single slide, just the main high yield points. Amino acids, are they macromolecules or micromolecules? Are amino acids macromolecules or micromolecules? Raise your hand if you think they are macro. No, they are micro. Protein is macro. So, you know, like I told you, you have to know your macromolecules and micromolecules. That's where how we break the bonds, endothermic, hydrolysis, and then the bond and the energy comes out. If I break a bond in catabolism and I get energy, is that exergonic or endergonic? Exergonic. Positive delta G. If the bond is broken, how is it broken? With an endothermic delta H plus or an exothermic? It's an endothermic. And an endergonic reaction. What's an endergonic reaction? An endergonic reaction is a reaction where the bonds are, coming back. So why are they coming back?
The heat is leaving.
Yeah.
The water's coming back and it's making the bond.
Excellent. So why do I do that? Why do I combine? Why do I
have enough energy?
Excellent. For storage. Excellent. I was looking for that word, storage. So when we want to store, we do anabolism. When we want to break down, we do catabolism. That's business. That's money. That's high yield. Okay. There are building blocks of proteins. There are essential amino acids. What are essential amino acids? I didn't ask you about those. The essential amino acids are amino acids that your body cannot produce. It needs from outside. But if it's non-essential, then your body is able to make them. We got to know the basic structure. This is money. This is business. The fact that we know what is like this one?
Yep.
Thank you. So you got to know your alpha carbon, guys. The carbon in the middle, and the carbon in the middle, that's the alpha carbon. And the alpha carbon must have how many terminals? Four different terminals. That's what makes the alpha carbon chiral or achiral? Chiral. So glycine, who happen to have an R group, who's an H, is it chiral now?
No.
It's achiral. So you got to know that one. That's very important. The alpha carbon is the carbon that's in the middle, right here. Rahim, right here. You see my mouse? You see where the mouse is going? That's the alpha carbon. It has four terminals: north, east, south, west. But it's not like that always. You could switch. I'm just saying it to remember it. The R group is the only group that is different among all the. So all the amino acids have four terminals, but they all share 3 out of 4. It is only that one R group that makes it unique and different. Except one amino acid happens to have an R group identical to the rest. What is that? Glycine. What is the R group that it has?
Oh, it has carbon or hydrogen.
Hydrogen, yeah. So it has double hydrogen. It has double hydrogen. So now that's a question on the exam. Draw glycine. It is not rocket science. You put a carbon in the middle, 4 terminals, north, south, east, west. And you put 3 out of 4 that everybody has. Hydrogen, amino, carboxyl, and then instead of the R, you put an H. And then you drew a whole glycine. Okay, so that shows us that this is the alpha carbon. That's how we look at it from a stereochemistry standpoint. And this is my alpha carbon. That's my carboxylic group. And that is my nitrogenous group. And this sulfur is what makes cysteine unique. So you have to know that cysteine is what enables us to make what kind of bridge? Disulfide bridge. Excellent. Okay, oh, you guys, a few people answering. I don't know why. Yes. Yeah. It's achiral because two of them are identical. Achiral, yeah. Achiral. Yeah, yeah. Excellent. Okay. You know these questions, you can find them on the slides, you can find them on the book, you can Google the answers for them. Okay, and you can find the answer and then confirm it with your book. So that could be a nice exercise. First of all, it's really fast. You know, now you put things on Google, they come up. So you put the answers, but not everything on Google is 100% accurate. Then that comes phase two, because you know you got to read things twice. So might as well do it first draft, then come back to your book and try to confirm your answer. Confirm your answer. Maybe something on your book that's not on Google that you're going to be tested on. But it's always good to do a run, a draft, a first draft. Okay, that's that. Now, if you look at cysteine, the R group for cysteine has sulfur, but everything else is the same. A hydrogen, an amino. The hydrogen has disappeared, but it's still there. It's invisible. We don't draw it if it's a hydrogen. That's the nomenclature, and that is the way we identify our organic molecules. You see, that's a carbon, that's an alpha amine, that's a alpha carboxyl. And who's that? What kind of amino acid is that? That is phenylalanine. So if we compare phenylalanine, it has 3 ports, just like cysteine, but the only difference is the R group. The R group for cysteine has an SH, and the R group for phenylalanine has a ring to it. Okay. D and L isomer. Almost all biological amino acids are in the L configuration. We have not talked about that. What else? I'm sorry? Yeah, it wasn't a study guide. So I don't want to disturb you. I want to make this a review session. You identify your carbo. So now if I show you a molecule like that and point to you with arrows and tell you, where is the alpha carbon in tryptophan? Look at tryptophan. Where's my alpha carbon? Is it this one? Raise your hand if you think this is the alpha carbon.
I told that because of the other. So I was actually gonna ask that because I was gonna just ask that. How are we supposed to know? Because I would think like it's not carboxyl.
Okay, so you see the carboxyl, that's the easiest one to rule out.
So no, but that's why I'm asking. Like, how would I know? I don't see a C, so I would just be like,
okay, if you don't see a C, where they connect, where you see a dot, that's a carbon. Unless when you see something like this, a dot and an oxygen, then that's an oxygen. But if you see just a dot, no atom around it, it's connected to oxygen, but the dot itself, that's a carbon. That's a carbon. The dent, that's a carbon. The bend or the dent, that's a carbon. Yes.
So you said that the alpha carbon is 4 capitals, right?
Yeah, 4, yeah. I told you the hydrogen, this is the way we draw organic molecules. The carbon, we don't put a C, we make a dent. We make a bend like that. That's a carbon. And the
You have to just remember that it's there because they don't draw.
Yeah.
Remember that it's there.
Yeah. So the carbon, we don't draw it when we're doing this kind of. So these are different conventions. The one on the top, we draw it like that, with electronegative really red, electropositive really blue. That's a convention. But the second convention is whenever you see a dent or a bend like that, that's a carbon. At the end, so sometimes even if this O is gone, something like, let's see if there is something like here, you see that end that ends with a carbon, but here you have an OH, so that ends with an OH, that's an O. This is a carbon, that's a carbon, that's a carbon, and that's a carbon. So how do you know the alpha? The alpha has 4 terminals. So look for the carboxylic terminal. That's connected to your alpha carbon. So that's my carboxylic terminal. That's my alpha carbon. My alpha carbon is connected to an NH2 right here. NH2 is connected to R. That's my whole R group. Where's my H? It's this. We don't draw it. If it's just an H, we don't draw it because otherwise we're going to end up with H here, 2 H's here, and H is here. We don't want the H's, but we know that they exist. We don't draw them, but because we know they exist. That's just a convention. So these alpha aminos, the amino side, is it the basic side or the acidic side? That's the basic side. That is the side that accepts hydrogen proton. So your amines, they are a buffer. So when you have suffering from acidosis, really excessive hydrogen proton, what does your body do? So your kidneys start to take a lot of hydrogen proton from your blood, throw it in the urine. So that means your urine is going to be really concentrated with hydrogen proton. You're breathing. Air is going to send out a lot of carbon dioxide because carbon dioxide through carbonic acid, it makes acid. So you're going to remove it. So your respiratory system going to remove, shunt out carbon dioxide, hydrogen proton. Remove out in the urine, hydrogen proton. And the proteins in your body, they have 2 terminals. This amino terminal, if you are having so much acidity, they will start to accept those hydrogen protons. So you have so much hydrogen protons, let's throw some in the urine, let's get some in the air, and we proteins, our amino terminal is gonna accept. That's why this NH2 will become NH3. But the acidic side is going to be the OH, so that whenever there is a drop in hydrogen proton concentration, you become so basic. When you become so basic now, what happens? Your kidney is not going to start to throw hydrogen proton in the urine. It's going to start to take hydrogen proton from the urine, throw it into your blood to increase the hydrogen proton because you have a drop in hydrogen proton. It's going to make your breathing level from hyperventilation into hypoventilation. And the amino acid is going to say, now it's the other way around. Now we don't have too many hydrogen protons to be taken by the amino terminal. Now we're going to have our hydroxyl terminal start to donate. So now this OH is going to become O minus. So these 2 terminals are like acid and base. So that's why I asked you on the study guide, which group tells me if my amino acid is acidic or basic? It's not the amino group, even though it's basic. It's not the carboxyl group, even though it's acidic, because those 2 are buffers. It is the R group that makes you basic or acidic. So now you see, now you can tell that is my alpha carbon, and these are my carboxyl group. Who is carboxyl group acidic or basic? Acidic. Is the NH2 acidic or basic? Basic. Excellent. Yes. I'm saying if there's going to be a question like that, it's going to be like that.
I'm sorry. Like, you see how you just asked, like, which is acidic, and are we just supposed to know that, or is there something you may have, like, helps us identify?
So, for example, if I gonna question something, it's gonna be like those arrows. Tell you, where is the alpha amine? The alpha amine is bounded to the alpha carbon. Where is the alpha carboxyl? This arrow. So, for example, I'll give you 3 arrows, tell you A, B, C. Which arrow points to the alpha carbon? This one. Which arrow points to the carboxyl carbon? This one. Which arrow points to the R group? This one. So you have to know the structure of the amino acid. That's your micromolecule. But if you're having trouble telling if amino acid is micro or macro, you have a lot of catching up to do, because we're asking questions way harder than that. So you have to go if you're having difficult times already the 4th week of school and people still not knowing what an amino acid is and what are the terminals are. You'll be asked on the exam questions about identifying and drawing, actually drawing glycine. Okay, so you have to pick up the slack. We haven't talked about the isomers. We will allude to them when we move on to carbohydrate. But for now, we're just dealing with this. So now you got to look at this molecule and be able to tell without any confusion, where is my alpha carbon? It's the carbon that is bound to my NH2 with my carboxylic and the disappearing hydrogen, and that's my R group. That's no longer a problem. So at high pH, meaning that when we are so basic, the OH is going to ionize, meaning that it's going to donate hydrogen proton. It's going to ionize. It's going to become minus. Because remember, we said that these two terminals, they act like acids and bases. So at high pH, who's going to, I already explained this. So these two terminals, when the pH is so high, that means the hydrogen proton concentration is high or low. If the pH is too high, that means hydrogen proton concentration is too high or too low. It means that the hydrogen proton is too low. So what's gonna happen is these guys are gonna come to help, and the OH is gonna throw away her hydrogen, and it's gonna become ionized with a minus electron. It's gonna ionize. But at very low pH, when there is so much hydrogen proton, this NH3 is gonna ionize. It's gonna become from NH2, it's gonna become NH3.
So then R group determines the
acidity and basicity. Yeah. If it's an acidic amino acid or no. So some amino acids are like alanine and glycine and proline. They are aliphatic group, and these are amino acids that do not engage in ionization. And these aliphatic R groups, they have neutral charges at physiological pH. What is physiological? I mean, that one you gotta know it. What is physiological pH? Physiological pH is 7.35 up to, so it's a range. It's not a number. It's a range, 7.35 up to 7.45. So these aliphatic R groups, if they are at neutral pH, or is the pH physiological pH, is it acidic or basic? Basic. It's a little bit basic. Excellent. So when you put an aliphatic group, look at the aliphatic group. Does not have anything does not have an ion. Does not have oxygen. Does not have nitrogen. So it will remain neutral. The other ones are gonna, you see, the minus charge is going to cancel with the plus charge, and you're going to be neutral. Now, when you look at the other groups, so these are the aliphatic. Don't forget that. The aliphatic groups are the groups that when you are at physiological pH, they are not charged. The charges cancel out with your amino terminal, who's positive. Which one is positive? The amino terminal or the carboxylic terminal? The amino terminal. That's the positive one because it picks up hydrogen proton. And the oxygen is the one who donates because it's the acid. The nitrogen accepts hydrogen. That's what makes it a base. What's a base? A base donates an OH or accept the H plus. And when it accepts an H plus, it becomes positively charged. It becomes positively charged. I don't know why are you guys laughing. And then you have to remember, please remember that which of the two terminals is acidic and why? It's the OH because it donates hydrogen proton. You are acidic if you donate hydrogen proton. And if you are basic, you accept hydrogen proton or you donate hydroxyl, OH, or you donate OH. Or you donate OH. But these aliphatic groups at physiological pH, the positive base is going to cancel with the negative oxygen, and you're going to have a neutral charge.
Because can you give us, can you give us an example of like a question you would give for that?
So I would give you one of these amino acids and tell you, is this amino acid allopathic? You say yes or no. 1 point. Why? 10 points. So then you say, oh, why? Because that it's at physiological pH, it's neutral because it only has 2 charges. The amino is positive and the oxygen is negative, minus and negative, neutral. This R group is not charged. That's what makes it allopathic. But we're going to look at other amino acids. They have a charge here. So when these two cancel, they're going to have an overall minus charge, or they're going to have an overall plus charge. Yes.
Can we equate allopathic to hydrophobic?
So yeah, you can do that, but not all of them, because you see this methionine has an S. So the S group, so with the sulfur does not engage in, is not an electronegative ion. It's not an electronegative ion. But most of them are non-polar group. Excellent. That's a good connection.
Professor, is this like, for example, like I see the study guide, you have 4 classes of amino acids. Okay, I don't see that word, so I'm just like.
So the 4 classes that you need to know are who's polar, who's non-polar, and when you get to polar, you got to find out who is basic and acidic. Excellent. Yeah. No, what do you mean negative bond? Yeah.
I know what she's saying, because when you're trying to study and understand all of this, you're kind of trying to like just make
Some of them
everything
it
make
says
sense.
like
Yeah, like in the charges, they have like a positive charge for the ones that are basic.
Yes, some of them are positively charged, and that is because the amino acid that they carry. So these are my allopathic. They cancel, not all the time, only when. That's the key point, at physiological pH. Okay. These are R groups that have carboxyl. So now, who's the R group here? The R group, what is the R group here? This one, because that's my alpha carbon. What is that? Alpha amino. And what is that? Alpha carboxyl. These R groups have an OH. These R groups have an OH. So a really nice question would be, which of the amino acids have an R group that is an H? Which amino acid has an R group who is a hydrogen, H? Glycine. Which amino acids have an R group that has an OH? These guys, acid. It's an acid with an OH. Yeah, if you have an OH, that means you're an acid. That's another easy one for you to spot out that the R group has an OH. Glutamic acid, aspartic acid, because of OH. Yeah, yeah. You see the OH? You show you the box with the OHs.
So the amino acid with the R group of OH would be those two right there.
Yeah, yeah, the aspartic acid and the glutamic acid. Okay, now these amino acids are not allopathic. They're not allopathic because at physiological pH, what happens? We already know it is not a secret that oxygen is what charge? Negative. And nitrogen is what charge? They cancel out. They cancel out. If it's allopathic, this guy is also like some methane or carbon. But with ascorbic acid or aspartic acid or glutamic acid, that O is going to become, what is the O going to become? O is always what? Negative. So if you have an OH, that tells you at physiological pH, when these two cancel, you're not going to be allopathic, you're going to be charged. And what charge are you? Negative. Going to be negatively charged. So that shows you the acids, yes.
Just for like nomenclature wise, because of all this stuff, they always put aspartate and glutamate. Can you put those words in the exam instead of aspartate?
No, aspartate is one thing. Aspartic acid is another.
Yeah.
Okay, we're gonna get to that. So the acids are on their own. Just for now, remember that O is gonna be negative and N is gonna be positive. At physiological pH, my terminal, my N, my alpha amino, my alpha carboxyl are gonna cancel out. Because who's positive? The amino. Who's negative? Or the carboxyl group, that's fine, the oxygen. Then they're gonna cancel out. Am I done? I'm not done because I'm not here. I was done here. I was done with this. With this. I'm done. Here, this guy and this guy cancel. That guy remains. You're done. You're allopathic. But here, you are not allopathic. Because when these two cancel, because you know in physiological pH, everybody ionizes. In physiological pH, that's O is minus. That's not a secret. Everybody know that. N is plus. That's not a secret. Everybody knows that. And then O is minus with just one, not two. One of them because they're going to have a resonance structure. So when you cancel minus and plus, there is an overall net charge of how much? Negative. So these are the acids and they are negatively charged at physiological pH. Now, look at serine. These are not but the easiest one to remember are aspartic acid and glutamic acid. Let's look at serine now. Serine, where's my alpha nitrogen? Right here. Where's my alpha carboxylic group? Right here. So when I ionize, what's this all gonna become? The oxygen is gonna become what? Charge. Oxygen is always negative. Nitrogen is always positive. Why? Why is oxygen always negative? Because it lost the hydrogen. It used to be OH. It lost the hydrogen and it got paid by getting a negative charge, electron. The currency for oxidation reduction is the electron. So it got an electron and lost the hydrogen. It used to look like that. You see where my mouse? Everybody, please look here. You see where my mouse? In the mouse, you had an OH. But at physiological pH, there's going to be deprotonation. The hydrogen proton is gone. It is gone. The hydrogen proton is gone. To increase hydrogen proton concentration to become acidic, and the oxygen was compensated by electron. That's why it became negatively charged, because electrons are negative. So it loses the hydrogen, is deprotonated, but it gains a negative charge. So oxygen is always minus. But the hydrogen proton, it used to have NH2. Remember, it used to be NH2 and it had 2 electrons. Now it's going to become NH3. The electrons are gone. So it becomes positive. But anyways, at physiological pH, they're going to ionize. When they ionize, O is always what charge? N is always what charge? And when they both cancel, you look at the R. Do I have no oxygen or nitrogen? I'm going to be allopathic. But if I have an oxygen, I'm going to have a net negative charge. Look at serine. You see serine has an OH. Threonine has an OH. So that tells you at physiological pH, that O is going to be minus, and that N is going to be plus, and that OH is going to be minus, so it's going to have an extra. You see that OH is going to have an extra minus charge. You see it's going to have an extra minus charge. They ionize at high pH. Okay, now remember cysteine. What is so unique that you need to know about cysteine? It is important for making what kind of bridges? Disulfide bridges. That's for the DNA that causes the DNA to cross-link. And it ionizes at a pH of 8. So it ionizes at pH of 8. So it gets a net negative charge. It gets a net negative charge. So if you put this amino acid in the pH of 8, the O is going to be minus, hydrogen proton is going to be plus, and the S is going to be minus. Okay. Now that's glutamate, different from glutamic acid. You see, glutamate is different from glutamic acid. Glutamic acid has an OH because it's an acid. It has an OH because it's an acid. That's glutamic acid, but glutamate is not an acid. So instead of the OH, what are you going to have? A nitrogen. You're going to have a nitrogen. You see? You're going to have a nitrogen. So if it had no nitrogen and OH, it's going to become glutamic acid. If it had no nitrogen and OH, that's going to become aspartic acid. Okay, you see arginine. There's arginine. What's going to happen to arginine? Let's figure that out. What's going to happen to arginine? The OH is going to be minus. The NH is going to be, they're going to cancel. And the N is going to give it a net positive charge. This is piece of cake. It's not. It's not. How is it not? No, guys. This is, look. No. I must speak for everybody. Please listen. We know that in our amino acids, we've been through this. Our amino acid, we have our alpha carbon. That's why we call the amino alpha amine, because it's attached to the alpha carbon. And that's why we call the carboxylic group alpha carboxylic. Then what's left is going to be your R group. If the R group is a minus, it's going to be minus, just like that guy, minus. If it has an N, it's going to be plus. If it has no oxygen or no nitrogen, just a carbon, it's going to have no charge, which makes it aliphatic. If it has an S group, it's going to become minus, but I'm not going to put that on this. But that's just for you to know that.
Put negative for the
So-
people, so you put negative.
Yes.
I want to make sure I understand. Can you go back to the new one?
Yeah.
All right, so for the R group, if it's O, which is oxygen, if it has oxygen, it's going to be negative.
Yeah, let me show you. Hold on. Let me step by step. Let me show you. If it has a OH, it's going to become, if it has an OH, negative. You see? Here it had an OH. And then when we ionize, this guy is negative, this guy is positive, this guy is negative. You see? This guy is negative, positive, cancel out. That's a whole net charge of negative. But if it had an N, keep going.
So wait, but all right, so my question is, can you go back, please? Whichever one. All right, so that one right there. That one right there. Are we going simply over the R group? So for example, you see how you have two O's, which mean oxygen.
Yes.
If both of those are negative, that cancels out. Right?
No, not both of them. One at a time. You cannot have double. So in the R group, you have a net charge. That oxygen represent resonance. So this double bond keeps switching between. You cannot have a negative charge on this oxygen because it's part of a double bond. It's already held by a double bond. It is the other free one. This one. See where my mouse is? You shoot the mouse. This one is going to ionize. Then the double bond is going to flip to this O, and the minus is going to go up to that one. But the whole charge is a minus.
So, okay.
Yes.
So just to clarify, it's the whole molecule negative or just the R?
The whole molecule becomes negative because of the R group. Because of the R group.
So let's say, for example, one of those, the R group was, and the whole thing would be a positive.
Yes. Because the other two cancel out.
And if no, then how?
Allopathic, yeah. Yeah. Very good. And if there's an S, it becomes a negative. That's just general. So we're not just dealing with O's and N's. We're dealing with S also. And what charge is S? Negative. Okay. And so that just shows us. Now this guy, so aspartic acid and asparagine, the difference is aspartic acid has an OH, asparagine has an NH.
So, just, so that one.
Yes, because it has an N. Look.
Yeah.
Let me show you the N group. So with the OH, you see the carboxylic OH is going to become negative. That's the O. Now let's look at the total N. Let's look at the N. That's the S. Yeah, right here. So now, are you following me?
Because he's focusing on the oxygen.
You're paying attention. So this one, you see where my mouse is? So now it's no longer O. What's gonna happen? What's gonna happen to the N? It's gonna become positive. So that N group is going to become protonated. And the oxygen, yes,
My confusion is for
asparagine.
I see a double bond in the R.
Yeah, yeah, you can have that as part of the R group. So don't think that the double bond is only part of the carboxyl. You can only have an oxygen with double bond only with the carboxyl terminal. No, you can have it in the R group too.
I understand that. It's like, how do I know to focus on the N instead of also taking them to
Because you see, this O is already committed. This double bond is already committed with this carbon. It's called a carbonyl group. So this, nothing gonna happen to it. It's the one who is free with one bond. This is double bonded. That does not engage in any reaction. So that's gonna be standard, like here. Let me show you. Like here, with aspartic acid, you see that one is committed. It's a carbonyl, or with two carbons. The extra one is the one gonna ionize. So if you put an N here, this one is stable. It's not gonna react. It's not gonna ionize because it's already held with two bonds. Is the other one? Yes. No problem.
So the double bond was an N, and that was a OH, it would be negative.
If the double bond here was an N,
That was
if the double bond is an N, it's committed with two bonds. It cannot ever. It's always the one who is not in a double bond.
so it's stable.
Yeah.
Yeah.
Yeah. Okay. Yeah. So you have to go and read the book, guys. You have to go and read the book. That's all in the book. This is not classified White House information. This is not political. This is not something secret. It's in the book. You have to go read it. To go read it. Okay. So then it's gonna fall. So, like you said, if we had an N, you had two NHs, the double bond is gonna switch. Are you following me? The double bond is gonna switch. One time here, one time there. If the double bond goes here, the charge goes there. The double bond goes there, the charge goes up there. But that all doesn't matter. What matters is your ability to distinguish who's your alpha carboxyl, that's a minus, who's your alpha amino, that's a plus. They both cancel out. Then identify the R group. Is it allopathic? It's going to be neutral. Is it has an O, like an acid, it's going to be minus. Does it have an N? It's going to be plus. Okay?
You're putting this on YouTube, right?
Yeah. Yeah. If you go and read the book, you'll get it faster. You will get it faster. Okay, what is this bond? So now when you look, now if we want to make a peptide bond, that's very important. If you want to make a peptide bond, what do? Hydrolysis or dehydration? If we want to make a peptide bond, we do dehydrate, we remove the water. You see the water? These are my two amino acids. 1, 2. So the OH is going to give us, because in order to have water, you got to have H2O. So somebody got to give me an O, and somebody got to give me the H's. So who gives me the O? The O doesn't come by itself. It is the OH of the carboxylic group. So the OH with one of the H's of my alpha amino. So if this is the alpha carboxyl, it has an OH, all of it will come out. And this alpha amino has one H, will come out as H2O. Okay, so that was amino acids. So this is the amino acids if we want to join them together. If we want to join the amino acids, bond them together. The bond that bonds the amino acids together is known as a peptide bond. The bond that bonds sugars together is known as glycosidic bond. And the bond that binds the DNA together vertically is known as phosphodiester bond. Phosphodiester bond. Yeah, that's very important. And the bond that binds the amino acids together is known as amino, is known as peptide bond. So how is it formed? By removal of water. Minus, remember I put minus H2O. And what happens is in minus H2O, O and the two H's are removed, and then a bond is made. The bond is made between the carbon and the nitrogen, and this is my green bond, my peptide bond. Okay. Now we're gonna go into, so we have to do, so we talked about amino acids and DNA replication, enzyme mechanism. These are all our topics, enzyme regulation, but we're not going to all of them, every single gene expression, hemoglobin, okay, and protein structure. So when it comes to hemoglobin, hemoglobin is really important. So let's do that one too. Make sure people understand the curves. You have to know your curves. Those sigmoidal curve and hyperbolic curve. I talked about it last week, but it's very important. So this oxygen needs to be transported, and diffusion is not fast enough. If your tissues rely only on diffusion for oxygen to be transferred into the tissue, that means it's not gonna get in time. Why do I need oxygen for my tissue? For oxidative phosphorylation and for aerobic respiration. So the diffusion is not fast enough. And sometimes when you're stressed out or you're running for your life, you're fight or flight, okay, you're gonna need hemoglobin to accelerate the passage. So hemoglobin, and you gotta know the difference between hemoglobin, myoglobin, and fetal hemoglobin. Those three are very important. First of all, I mentioned to you already that if we want to know the difference between things, we have to first know the similarities. What are the similarities between hemoglobin and myoglobin? Both of them are proteins. Is one protein the other carbohydrate?
No.
Is one protein the other one lipid? No. So all of them are proteins. But the difference is right here. Hemoglobin is quaternary. It has 4 subunits. 2 alpha, 2 beta. 2 alpha, blue, 2 beta, red. And on each subunit, you have a heme group, ferrous, Fe2 plus. That is the seat of the oxygen. The oxygen sits on the heme group, and the heme group is on the subunit. So if hemoglobin has 4 subunits, how many heme groups does it have? 4. And how many oxygens it will have? 4. So when oxygen is unloaded and carbon dioxide is loaded, how many carbon dioxide I'm going to pick up? 4. So we're just going to replace, the heme is going to drop. It's going to go from the ferrous state to the ferric state, Fe plus 3. So from Fe plus 2, ferrous, that's when we are bound. Ferric when we let go. Myoglobin. Myoglobin is 1 subunit with 1 heme, 1 O2 with 1 oxygen on each one. Okay? Now, myoglobin has only 1 subunit, but it is tertiary. So how do we go from primary to secondary? By hydrogen bonding. The hydrogen bonding is what's going to help us flip on our back. So these amino acids have a hydrogen. We talked already about the bonds. Some are electronegative, some are electropositive. So the hydrogen is going to bind to the oxygen with really weak bond known as hydrogen bonding of these different amino acids. That's going to cause the primary structure to become alpha helix, and that is known as secondary structure. And the alpha helix is going to turn into a Or we can excellent either have alpha helix or beta sheet. But if I want to go from secondary alpha helix and beta sheet into tertiary, what do I use? Hydrogen bonding, more extensive, but now not just that, that's not enough. Relying only on hydrogen bonding to go from secondary to tertiary, that's not enough. Now we need an additional form of bonding known as hydrophobic interaction. Hydrophobic interactions are not enough. We need van der Waals interaction. So we need the three of them. We need hydrogen bonding, we need van der Waals interactions, and we need hydrophobic interactions. And then I made it from my beta sheets, like Kareem said, and my alpha sheets, like Agatha said. Now I've made it up from second level to tertiary. Now I am myoglobin. I am myoglobin now. If I want to become hemoglobin, if I want to flip now from third to fourth, hydrogen bonding is not enough. Van der Waals are not enough. Hydrophobic interactions are not enough. What else do I need? Ionic bonding. That's going to now take me to the next level. So hemoglobin is quaternary. Myoglobin is tertiary. Some people say it's one subunit. Yes, it's one subunit, but it's folded 3 times on itself. People think that hemoglobin is quaternary for myoglobin is one subunit. That means because it has one. No, the subunit is folded 3 times on itself. Because you can tell myoglobin does not look like an alpha helix or a beta sheet. No, it looks like many alpha helix folded upon themselves. There's a prosthetic heme group. That's the yellow group. And we know that's the ferrous. That's the only exam. Ferrous Fe plus 2. That is the form that binds oxygen. Ferric Fe plus 3. That does not bind hydrogen. So that is your ferrous group to bind oxygen. Ferric does not bind oxygen. No, that's that form that does not bind oxygen. So for example, that shows you that hemoglobin is excellent for transporting oxygen and carbon dioxide, transporting oxygen. But myoglobin is not good for transportation. It's good for storage. So we just store oxygen on it. So attach the remainder of the global subunit. Okay, now we're going to talk about the cooperativity. That's the form of a positive feedback. We began this course with negative feedback and positive feedback. We said that in positive feedback, in platelet plug formation, and we're making a thrombus. If we are injured, one platelet stick, the second platelet on top of it, the fourth platelet on top of it, the fifth platelet on top of it. That's positive feedback. The more stick, the other stick. In hemoglobin, the same thing. And we call that cooperativity because if one oxygen binds, the rest will bind as well. So look at here. That's hemoglobin. In the T state, T means you are inactive. You really had a nice mnemonic last week, Agatha. What was it? T is for what? And R is for what? Relax. I'm sorry, can you say it again?
When we went uptown, downtown.
Oh, very good. Excellent, excellent. So to help you remember T state, because you got to know those. You got to know what the T state is. The T state when it is tense, or like Agatha said, top, because that happens at the top. When you're in the lung, you are about to load the oxygen. So look at what happens. When one subunit binds, the other 3 will help it. Like my 4 aunts, when my mom got married, all of them got married after her. All of them first, they said, no, they don't want to get married. They want to do something. I don't know, business or education or something. My mom got married, they all get married. So that's exactly what happens. Here, when one bound oxygen, the other 3 bound oxygen. That is cooperativity. So one binds, the other one's bind. That is known as cooperativity. That's positive feedback. Just like the platelet, if one platelet binds, the second one will be on top of it, third one will be on top, fourth one will be on top. No, that is just T means you have no body, no oxygen. You're just empty. You're tense. Like I like what she said at the top of the lung. You just arrived to the lung. You're carrying carbon dioxide, no oxygen. You are at the top. You made it because you know we had a downtown trip and an uptown trip. Uptown, you're at the lung. You have so much oxygen, but no, you have no carbon. So you have carbon dioxide and no oxygen. So you reach tense top. So you let go of carbon dioxide. One oxygen bind. You see one square turn into a circle that bound oxygen. Then the second one bound oxygen. Then the third bound oxygen. You see, the first one became bound oxygen. Next, two now have oxygen. Third, the third has oxygen. Fourth, the fourth has oxygen. When you are fully oxygenated, you're R now. I don't know if it's relaxed because now both of that is happening at the top. I got it. So it's not like the T is happening at the top and the R is happening in the bottom. No, both are happening at the top. So when when the hemoglobin gets there, it has no oxygen. That's T. When it unloads all the carbon dioxide and picks up oxygen with cooperativity, one binds, the second one binds, the third one binds, the fourth one binds, then it becomes R. So T and R are one. T means no oxygen. No oxygen, not saturated with oxygen. And R means you are fully saturated with oxygen. You see? T and R. This graph, oh my God, this graph is so important. I drew it for you last week, remember? I wish I had recorded it last week. This is really important. What does this? You're never going to understand a graph without understanding the coordinates. Who's the x-axis? Who's on the y-axis? Who is the independent variable? And who is the dependent variable? So the independent variable represents for me the partial pressure of oxygen. And that represents for me the saturation of my molecules with oxygen. So this graph shows you that hemoglobin will let go when there is low partial pressure of oxygen. It has no saturation. It doesn't make sense for hemoglobin to have to be saturated with oxygen in a place that has no oxygen, like in tissue. A place suffocating need oxygen for oxidative phosphorylation, for aerobic respiration. So oxygen will oxygen saturation of oxygen in hemoglobin. So hemoglobin is not going to be saturated with oxygen in a place that has no oxygen. But when you look at myoglobin, it's really saturated. So for example, look at a saturation of 10. I'm sorry let's look at a partial pressure of oxygen with 10. So you only have 10%. To make it easier for you, think of this x coordinate as 10% oxygen. There's only 10% oxygen there. When there's only 10% oxygen, hemoglobin will only have 10% oxygen, almost 10% oxygen, because it let go of the 90%. Because there's only 10%, it let go of everything. But look at hemoglobin. Hemoglobin at 10% almost has 75% oxygen. So it didn't let go. Did not let go. At partial pressure of 25, at a tissue that has only 25% oxygen, hemoglobin is only having 40%. So that means it let go 60. Look at myoglobin. It has almost 90%. So this sigmoidal like a S shape. This green sigmoidal S-shaped hemoglobin curve shows you that the lower the partial, we don't say percentage. I said percentage to help you understand, but we don't say percentage, we say partial pressure. So the lower partial pressure of oxygen, the lower percentage of oxygen, the lower saturation of hemoglobin with oxygen. Okay? Your mom is not going to have a lot of food and you're hungry. When you have low food in your stomach, your mom is going to have low food because she's going to give it to you. But myoglobin is not going to do that. It's going to hold on to it, tell you, deal with hunger. Maybe you need it later on, and we'll give it to you. So hemoglobin is going to always be, when the tissue is low in oxygen, hemoglobin is going to let go. It's going to be low too. But myoglobin is not. This is a S shape. The green one is hemoglobin. That's S shape. And the blue one is myoglobin. So what does it mean when the curve moves to the right? When the curve moves to the right, it means that I have more partial pressure of oxygen. So that means I do not need hemoglobin to unload. So that I'm not exercising. When I'm exercising, I shift left. I shift left when I'm exercising. So now my graph is going to shift left. So I'm going to unload oxygen I'm going to unload a lot of oxygen at the low partial pressure. But when I shift right, it means that I have, you see 100. 100 is on the right. 100 means I have a lot of oxygen. So now hemoglobin will have a lot of oxygen. If you have a lot of oxygen, hemoglobin will have a lot of oxygen because he's not going to give it to you. Hemoglobin will only have low oxygen if you have low oxygen because he's going to give it to you. Does that make sense? All this got to make sense because it's going to be on the exam. Very important, guys. Very important. It's very confusing because students think of saturation. They don't know saturation of what? This is saturation of hemoglobin with oxygen. So hemoglobin is carrying oxygen. It comes to a place where there is a lot of oxygen. Is it going to be saturated with oxygen or is it going to be unsaturated? It's going to be saturated because there's a lot of oxygen needed. But when you're exercising, no oxygen. Is it going to be saturated? It's not, because it's going to let it go. That's what happened here. The mmHg, I told you to make it easy, think of it as percent of oxygen. So that's the tissue. Tissue is here. Hemoglobin is here. That's what makes this curve confusing. Because O2 is representing hemoglobin. Maybe I should have added a title here. That's hemoglobin oxygen concentration. And that is tissue oxygen concentration. So at low tissue oxygen, what does that mean? It means I'm running a marathon. My tissue is suffocating. No oxygen. Then hemoglobin is going to have, you see the green? It's going to be low too because it let it go all of it. But here, 100%, when I'm just hanging out, I'm chilling. I'm just watching the NFL. So here, my oxygen is 100%, and so is my hemoglobin. You don't need. But look at myoglobin. At low saturation of oxygen, hemoglobin has low saturation. But myoglobin, I'm sorry, at low saturation, hemoglobin has low saturation of oxygen, but myoglobin is holding on to it. You need it, it's holding on to it. It's not going to give it to you because it's not there to transport. It's there for storage. Okay? Yes, sir.
So when does it let go?
It doesn't let go. It's going to stay like that. We use it as a reservoir, and hemoglobin will take the oxygen from it. So it's going to support hemoglobin. Green is known as what? Hyperbolic or sigmoid? That's sigmoid. Blue is hyperbolic or sigmoid? Hyperbolic. Who is hyperbolic? Hemoglobin or myoglobin? Who is sigmoid? Hemoglobin or myoglobin? Who is better for storing? Hemoglobin or myoglobin? Who is better for transportation? Delivering? Oxygen or hemoglobin? I promise you all of that on the exam. Every single thing.
Wait,
I cannot do that. I cannot. No, that's not how it works. You cannot do that. You have to pay attention. There's a lot I got to cover. Yeah. Okay.
for travel and all that other good stuff, that's the hemoglobin. Alright, cool.
Yeah. No. That's different, Kareem. Kareem, we're not talking about the T and the R state. We're talking about this curve. There's another one coming up too. That one is that one you should see it in a little bit. This one is part one. You should see part. We're gonna see part two in a little bit. Yes. Hyperbole. It's blue. You see, you can see a hyperbola. You see the hyperbola like a bend?
Oh, okay.
Yeah, the blue. Okay. Okay. All right. Now look at the other graph that you gotta know. Guys. The other graph that you gotta know is the Bohr. That's graph one. Then there is graph two. Okay. So no, this is not as complicated. I'm gonna show you in a little bit. So here, now we're not just talking about that graph, that old graph here. Here we're just talking about oxygen, if there's a little bit or not. But remember, a suffocating tissue is not just an issue of oxygen. Let's say you have all the oxygen in the world, but so much acidity. You have all the oxygen in the world and excessive carbon dioxide. So oxygen in the world and excessive nitrogenous waste. So much oxygen, but there is 2,3 BPG, 2,3 biphosphoglycerate. So now that shows you there's additional stuff. So you're in school, you're not just paying tuition, you're paying tuition, transportation, room, board, so other stuff. So the tissue is not just suffering from whether oxygen is here or not. There's other things that's gonna make the oxygen demand even more than just it's not there. So for example, if oxygen is not there and there's no metabolic waste, but if there's oxygen is not there and there is metabolic waste, am I gonna demand the same amount of oxygen?
No.
Now my oxygen demand is gonna double, not because I don't have oxygen, but because now I have waste piling up. What if now I don't have oxygen? I have metabolic waste and acidity. Hydrogen proton increases. That's gonna make my demand for oxygen even more. So that Bohr guy, he's the father of Niels Bohr, the guy who remember when we discussed the atom, the isotope, one has neutron, one has proton. That was Niels Bohr. This is his father. That's the father. So now the father, he did this. He said, this graph, protons bind to hemoglobin. Protons change the hemoglobin shape. The reshaped hemoglobin lose oxygen. The rapidly metabolizing tissue release protons. Rapidly metabolizing tissue get more oxygen from hemoglobin. So that shows you that does hydrogen proton bind to hemoglobin? No, it just displaces it, but it doesn't compete with it. It just displaces it. So that shows you if I am hemoglobin, I'm carrying oxygen. This hydrogen proton is going to kick the oxygen out, and the oxygen is going to go to the tissue. So this graph is telling you, it's going to tell you that as you go and it will show you that if the demands multiply, so now you're not only dealing with a shortage of oxygen, now you're dealing with shortage of oxygen, metabolic waste, and hydrogen proton concentration. You will now, as hemoglobin, lose more oxygen at the same partial pressure. Like for example, if you're just dealing with a shortage of oxygen, let's go to the old graph. If you are just dealing with oxygen at 25, you will be having, for example, 35% oxygen, and you will let go 65. But at this is so important. At this same level, if now you're not only dealing with hydrogen, with oxygen loss, you're dealing with hydrogen proton pumping, piling up, nitrogenous waste, now you're gonna be saturated even less. So at 25, you're supposed to be you're supposed to be 35% loaded, the graph is gonna shift to the right. So now you're gonna be only saturated with 20. Because now these extra demanding, negative, stressful conditions has led hemoglobin to lose more than in what it would if everything was okay. You understand? So you spend this amount of money. When COVID came, did you have the same spending or did it double? It doubled. So the same thing here, when we just dealing with there's no oxygen at, for example, at 25%, hemoglobin would let go. At 25 millimeters of mercury, hemoglobin will let go of 65% the oxygen that it has and will only have 35%. But if it, at the same condition, now you add hydrogen proton, now you add nitrogenous waste, now you add metabolic waste buildup, what's going to happen is at the same 25, it will not just let go of 65 and keep 35. It's going to let go of 75 and keep 25. So I will let go more to keep up with that extra stress. So that made my graph shift left or shift right.
Right.
You got to shift right. Because you can tell with the math, if this point here, and now you gotta let go of this, then your graph is gonna look like that. So it shifted to the right. So bad things make us shift rightward. Good things, when you're now refreshed, when you're running the marathon, you shift that way. When you're now refreshed, you shift always left. So you shift left when good things are good. You shift right when the nightmare. Okay? So don't look at all these graphs, they tell you the same thing. Okay, orange demanding more. So that shows you less oxygen bound at the same pressure. That's what we were saying. At the same pressure. So what does it mean when the graph is shifting to the right? It tells you that at the same pressure, where we have an expectation that at this pressure, hemoglobin should unload of this and be saturated with that. But when you add these dangerous stuff, hemoglobin now is going to have to accommodate. That's an accommodation. So it's going to lose more. Yes?
So basically, like each line is just saying like, okay, now you add this, now you add this, now you add this, now you add this.
You got it.
Same concentration.
Yeah. Yeah. So at the same, hold on. So at the same concentration, are we saturated more or less?
Less.
Less. Excellent. Because
you have to give up more for the increased demand.
you got it. You got it. Yes. Yeah. So when things are, is not just running. I gave an example of running. What about if you are emotionally stressed? That's an equal problem. So the thing is here, when things that are not just, so this graph represents for us how much hemoglobin is saturated with oxygen at times of how much oxygen is saturated with, how much hydrogen is how much hemoglobin is saturated, how much oxygen is saturated, how much oxygen is saturated, how much hemoglobin is saturated, how much oxygen, how much hemoglobin, I've been doing this since 8 a.m. How much hemoglobin, how much of hemoglobin is saturated with oxygen. So when demands increase, are you going to be saturated with more oxygen or less oxygen? Less, because you have to give up more to keep up with the demand. You're going to spend more. Okay? You spend weekly this amount of money. Now you have a guest. You're going to spend the same? No. You're going to spend more. So now with dealing with just oxygen, you're going to be saturated with 35% at that pressure. But when there's metabolic waste and there is demand and there is on top of it, then you're going to be saturated even less because you got to give up more oxygen. You're going to shift right. You're going to shift right. So the acid favors release of oxygen. Carbon dioxide favors release of oxygen. Acid and carbon dioxide are released by rapidly metabolized tissues. So I told you what you have to be able to recognize what does it look like to be a nasty tissue, a tissue that is suffering. It's gonna have low oxygen, it's gonna have low pH, excellent Agatha. It's gonna have high carbon dioxide, it's gonna have high 2,3 biphosphoglycerate, bisphosphoglycerate, 2,3 BPG, excellent. It's gonna have high acid. Yeah, excellent, high acid. Yes.
To be clear, when it moves on the graph right, that's lower oxygen acidity.
That's acidity, saturation. So, okay. Your name is Linda. Lydia. So now, no matter what you say, I'm gonna say the brain is now shut down. Okay, Lynette. Oxygen is there to do what? It's there to deliver oxygen, not to everybody. It's a discrimination. If you need it, it will give it to you. If you don't need it, it will not give it. Now there is, like, let's say, and we look at it in a confusing way. We don't say how much you give, we say how much you have. But we use that in reverse to calculate how much you gave. So if I'm hemoglobin, I have 100%. Now I have 25%, it means I lost 75% of my oxygen. So that graph is telling you when the tissue saturation with oxygen is low, hemoglobin is gonna give up all the oxygen and it's gonna be low too. So both are low. The tissue is gonna have low oxygen, hemoglobin is gonna let go of 80% of the oxygen, it's only gonna have 20%, now it's gonna be low too. But when the tissue has high oxygen, hemoglobin can say you don't need it, so instead that has 80%. But this is another issue now. When the tissue is low, for example, we know at this particular pressure, we studied hemoglobin, that's the graph. We know at partial pressure 20, you're supposed to have a saturation of hemoglobin is supposed to be saturated with 35. That means it gave off 65. But then when they studied, so now it's not only just there's no oxygen. Now there's no oxygen, the heat is up the roof, there's so much acidity, there's so much metabolic waste building up, nitrogenous waste, 2B3, biphosphoglycerate. These are 7 different piling up, more demand, right? So we have to accommodate that. Okay, I cannot just be giving the same amount of oxygen to the tissue at the same pressure who's not dealing with all that kind of stuff. That's an additional demand. So now I have to lose more. So my curve is going to shift left to accommodate. Shift right. Very good. What we done? You're going to shift right. You're going to shift right to accommodate for that. I usually, at this pressure of oxygen, I'm supposed to be, you are pressure 20, I'm supposed to give you 65%. But now, you are not only dealing with just 20, now it has metabolic waste, I'm going to give you extra, so now you're going to lose more. So when there's more demand, you don't get the same amount. Does it make sense? It makes sense if you just have low oxygen, I'll give you this amount. But you have the same amount, and then you are suffering with more metabolic waste, more nitrogenous waste, more carbon dioxide, then I have to accommodate for that. Does it make sense? Yeah, you have to you gotta go.
So
Yeah, it's good.
tissue has low oxygen, then hemoglobin will be low.
Why? Why, Curry? Why is that true?
I was asking if that was correct.
Yeah, but you should think about it. A tissue that has low, who's carrying oxygen? Hemoglobin. If the tissue has low oxygen, what do you think the hemoglobin is going to say? You have low oxygen, you go die? It's going to give it. So it's going to give it the oxygen, it's going to become itself low. Your dad sees you have no money. He's going to have no money too, because he's going to give you. He has $100 in his pocket, you have no money, he's going to give you $100, or maybe give you 50. Now both of you are going to be low. Same thing with hemoglobin. He's carrying oxygen. You have no oxygen. So at this, for example, think of it this way. At the end of the month, your salary is 3,000. So if you work these hours, you get 3,000. As days went on, COVID hit, for example. So now there's additional problems. So now you're not supposedly at the end of the month you're supposed to get 3,000. But now you're not just dealing with a regular job, there's extra demand, so you're gonna get 3,000 plus what did I check? The check. I was in med school out of here, I didn't get it. But what was it called? Stimulus. So you're gonna get extra because you're gonna deal with extra. That's exactly what happens there. The hemoglobin is gonna come and say, usually at this amount, you're gonna get this amount of oxygen. But now you are different from tissue in the abdomen, you're different from tissue in the brain, you're tissue of the legs, you're running a marathon. So you're dealing with extra stuff. So I'm gonna give you what you usually get at this level of low oxygen, plus even more because there is more metabolic waste generated. Okay? You gotta have to read the book. And the book, you know all of it, there's regions in it that are corresponding to the study guide. What would you think that?
That's what I heard.
No, you see, you hear people, they take you bad places. Okay, don't listen to the people. Don't listen to the people. And don't listen to the people when they tell you something about somebody. Okay, anyways. Listen to me when I tell you something, that's good, but don't listen to the people. Anyways. Okay, hemoglobin. That was 80% of the questions of hemoglobin. That's you really gotta know that one. Okay. So why do we need, and you need to know your R state. R state is when I'm full of, it doesn't mean I am tight. That's wrong. It doesn't mean I'm top, because both are at the top. When I'm entering the lung, I'm at T conformation. When I'm leaving the lung, I'm at R conformation.
Because you're filled up with that.
Yes. Okay, hemoglobin at the movement of oxygen. So rapidly metabolizing tissue produce acid. You got to know those. Those slides that I stopped at, you gotta know those. 2,3 BPG, big concern for okay, smokers. That's part of the study guide. So smokers, they have a lot of 2,3 BPG. So their oxygen, their hemoglobin does not get fully saturated because 2,3 BPG displaces it. This slide is really important. I made a couple of questions about it. Okay. Okay. It's gonna be on the thing anyway. What's happening? Why is there a French Revolution? What's happening? Okay. Can everybody please pay attention? This is very important. What does this say? This says that 2,3 BPG, 2,3 bisphosphoglycerate. You don't have to memorize the whole name, but you gotta know that 2,3 BPG is negative toxic metabolic waste. It is a big concern. Excuse me. I'm sorry. This is the second time I tell you that. You are talking and you are disturbing me. Please stop with her. Please ask me. I will answer you. No fear. Just raise your hand. I will be happy to walk over there and explain it to you. But don't ask her. Ask her later in the discussion in the library. But please don't disturb me as I'm explaining. This slide is on the exam. 2,3 BPG is a big concern for smokers. So the blood of smokers has high levels of 2,3 BPG. Not a secret, not unexpected, because smoke, nicotine, stress, damage, and metabolic waste. That one did not need a lot of thinking. That the one is clear. Hemoglobin gets locked in the T state. So we already said that hydrogen proton is going to displace oxygen. We want that. The 2,2-BP3 is going to displace oxygen from hemoglobin so we can come off to the tissue. But the problem now, this 2,3 BPG is not in the tissue. It's in the blood. So now the hemoglobin didn't even get to the blood. and 2,3-BPG is pushing it out in the blood. So it arrives to your tissue to remove carbon dioxide, but it's not bringing in oxygen. So they always have low level of, so they breathe, but it's not going nowhere. It's not going to the tissue. Oxygen carrying capacity of the blood is reduced. I already talked last week about carbon monoxide. What did I say, Kareem, about carbon monoxide in hemoglobin? It is a competitive inhibitor. It not only displaces, it displaces and it sits on the place of oxygen. So now your hemoglobin is not carrying oxygen, it's carrying poison. So it's delivering to your tissue, so it comes to the tissue. Usually it says, tissue, here's oxygen, here's oxygen. Now it's gonna say, here's carbon monoxide. So now it's killing you, it's bringing your enemy. Is because carbon monoxide binds, and in fact, unbelievable, carbon monoxide binds hemoglobin more than 10 times stronger than oxygen. So that's why carbon monoxide poisoning is very dangerous.
Why is it, you know why they have red lips?
I'm sorry?
Why they have red lips?
It's called pursed lips. That is because when that is because the smoke. These are the smokers. Talk about smokers.
Oh, no, but monoxide poisoning gives cherry red lips.
Cherry red, maybe, but I just
No.
remember the pursed lips
Oh.
with those who
Yes,
smoke
smoker.
because the oxygen. So these tissues of your lips, they are different from the tissues of the rest of the body. So they are very, very sensitive. So smoke in your skin is not going to do as much damage to your lips. It's going to destroy your lips, make them burst, and make them but I have to look into that. It didn't come to me. Why do they have pink lips? Okay. All right, you don't need to know that. This is not on the okay. Okay, so this is not even on the exam, but it may help you with something that you can answer, and that is an additional histidine is present at the heme iron site. It reduces the affinity to carbon monoxide, but it does not eliminate it. Carbon monoxide in cigarette smoke. There's carbon monoxide in cigarette smoke, and that carbon dioxide does not bind to heme, nor do proteins. So how about, nor do proton? How about I tell you something really nice? How about I ask you on the exam a question? What is the amino acid? The amino acid that is found in hemoglobin? Aspartate, aspartic acid, histidine, or glycine? What are you gonna say? Histidine. So that's the one amino acid that is really, working with this oh boy. The body makes different globin over time. Most variations are centered on okay, so now this is fetal hemoglobin. Okay, now look at this curve. Here, myoglobin, hemoglobin. If fetal hemoglobin is pink, what do you think the pink curve? First of all, do you think the pink curve for fetal hemoglobin, do you think it is sigmoid or hyperbolic? It's hemoglobin, whether it's fetal or adult, it's hemoglobin. So it's definitely gonna be sigmoidal. Now, is it going to be in front or is it going to be behind? And think about it. What does in front mean? What does behind mean?
Behind. I mean, front.
Who thinks is going to be fetal hemoglobin going to be in the front?
Oh, no.
Who thinks fetal hemoglobin is going to be in the front? Front. Who thinks it's going to be in the back? If you think it's going to be in the front, raise your hand. No, I told you when a graph shift right, what it means. It means things are bad with accommodating, but what that means what? That means I let go easily. Guys, Lynette, that's you. How did I pick that up? I thought I shut down after 6 p.m. I picked that one up too. I don't internalize anything after that. Anyways, when I shift right, it means I accommodate. But what does it also mean? It means I'm not tight because I let go. So if fetal hemoglobin is in front of green and blue, it means that it is gonna let go easier than hemoglobin. If it's gonna be behind hemoglobin, it means it's gonna let go, but not as easy as hemoglobin, gonna hold on to it. If it's behind myoglobin, it's gonna definitely hold on more than myoglobin. And obviously, it's impossible for fetal hemoglobin to be behind myoglobin because it's not a store, it's a hemoglobin. It's for transport, and then it's gotta be in front. But how front? Is it front even more than hemoglobin or behind hemoglobin? Yes, more. It's gonna be more. So it holds to less amount of oxygen than hemoglobin at the same concentration. Why do you think so? So if fetal hemoglobin shifts, if fetal hemoglobin's curve is in the front, that means it's gonna let go of oxygen more loosely. It's gonna be more efficient in letting go of oxygen than hemoglobin. And if it's behind hemoglobin on this curve, then it's gonna hold on more to it. That's why if you hold on more, you're all the way in the back, like myoglobin. If you hold on, if you let go, you're gonna be in the front. Now we're done with adult hemoglobin. That's green. We're looking for fetal hemoglobin. What do you think it's gonna be? So he said it's gonna be in the front. He's gonna let go more oxygen. Why do you think that?
I think because in the baby they have more stress, so that would make the fetal hemoglobin let go faster.
And what makes you think that babies have more stress? Well, guys, that's very important.
Not in the stress we think of, but like they're not breathing by themselves.
So if they're not breathing by themselves, they are inside the mom, you mean?
Yeah.
Then the mom is breathing on their behalf. So all of their red blood cells are getting saturated. So that when the mom goes, she's giving oxygen to her own red blood cells and the red blood cells of the baby.
That's why she's sleeping.
I'm sorry? You can say something, then never mind. So can you say what? Somebody say something so I can work with it. He just said something and he said the baby has more stress. So now we're discussing why do you assume that the baby is under more stress than the adult? He's not as developed. Does that mean, you mean like because it is developing, it's demanding more oxygen? Yes.
If we're talking about stress, and you just explained that the mother's treating for both, wouldn't the baby be less stressed?
Exactly. It's the adult who's more stressed than the baby. It is counterintuitive. The adult is dealing with more stress because it's a more body, the bigger body. That's a little tiny body. That's feeling longer, and the baby does not consume, definitely, per tissue, the baby does not consume as much oxygen because the baby drinks milk. It doesn't need metabolism for fat and for protein and for steak and for, all that stuff. It has a small system. It's satisfied with a little bit. Just give it breast milk, he's happy. But the human guy, the adult guy, he's dealing with a lot. You understand? So the baby fetal hemoglobin is going to be behind.
It needs less.
You see? That's the baby, the red. So it has fetal hemoglobin can't bind to this 2,3-BPG and remains all the time in the R state, saturated with oxygen. So the hemoglobin is saturated with oxygen, but it is not susceptible to 2,3-BPG. So that's what the body makes different globins over time. Most variations centered on birth. But fetal hemoglobin, this is so important. The adult hemoglobin is 2 alpha and 2 beta chains, but the fetal is 2 alpha and 2 gamma, and it's right in the middle. It's definitely better than myoglobin. Otherwise, if it's behind myoglobin, that means it's not for transportation, it's for storage. But it's a hemoglobin, so it's going to be in the front. And it cannot be hyperbolic. It has to be sigmoidal, but definitely not as sigmoidal as because as adult hemoglobin, because adult hemoglobin is very sensitive. Adult hemoglobin is very sensitive to acidity. Acidity will make it let go of oxygen. 2,3 BPG, let go of oxygen. Nitrogenous waste, let go of oxygen. Carbon dioxide, let go of oxygen. But fetal hemoglobin, not as sensitive. Yeah, all of these kind of stuff, yeah. Sickle cell anemia. This is very important. The single nucleotide polymorphism. That one is on the exam. I'm going to ask you on the exam, what are the 2 amino acids implicated in sickle cell anemia? And you're going to say valine and glutamic acid. And I'm going to tell you exactly who are we supposed to have to be a normal. To be a normal is glutamic acid. What happened? A mutation, SNP, single nucleotide polymorphism, led to a mutation that changed glutamic acid into valine. Yes.
It's multiple choice, right? Was your questions or?
No. There is short answer too. So it's a written exam and there's short answer. Yeah. There is multiple choice and there is short answer. Honestly, I haven't, but be ready. And be ready, yeah.
So for the short answer, is it like 7 of them? 6 of them?
No, that's not the way. We are here to learn. We're not here to like economically say, okay, how much study should I do? We're here to appreciate.
I'm trying to figure out like, you know what I mean?
We usually don't get your answer questions.
Yeah, that's what I'm saying.
Yeah.
Yeah.
This is the first time.
Yeah. Yeah, especially for this one. Okay, guys, another thing I forgot to tell you. Do sickle cells sickle all the time? No. So a sickle cell patient who is relaxed and a normal patient who is relaxed, you take blood sample for both. Can you tell who has sickle cell anemia? No. So the sickle cell will only sickle under what? Stress, like when there is metabolic demand or exercise. So the shape change happens in low oxygen conditions and exercise caused by polymerization. So these rounded cells move easily through the capillaries. If you are rounded, you move easily. But if you are sickled, you get stuck. When you get stuck, who destroys red blood cells? The macrophage. And that makes the macrophage. That makes the macrophage. I'm sorry, not the macrophage. The spleen, the graveyard. The spleen is the graveyard. Okay. Okay, another thing about the malaria. Sometimes you get an advantage. So sickle cell anemia is a homozygous recessive. 2 little R's. So if you have 1 big R, 1 little R, you're a carrier. If you have 2 big R's, you're normal. You don't have sickle cell anemia. If you have 1 big R, 1 little R, you are a carrier. If you have 2 little R's, then you have sickle cell anemia. Who has an advantage? The heterozygous. Yeah, excellent. Benefit of sickle cell mutation for heterozygous. No benefit to the homozygous. Okay. And
the benefit is?
The benefit is confers immunity against, you never get malaria because now the mosquito, the parasite that lives in the mosquito's belly, the mosquito's stomach, when it just like the HIV virus, want to live inside your white blood cell. The malaria, the parasite, you want to live inside your red blood cell. But if it's heterozygous, it cannot enter. Same thing with HIV. HIV, they found out that some people have a mutation that on the surface of the cell, you will have, there's an accessory receptor in addition to the big receptor the virus binds to that helps the receptor enter. The people who have a mutation, that second accessory receptor is absent, they will never get HIV. protein structure. Protein structure, the sequence, we said that sequence is a linear chain. What caused us to go from a sequence that is primary to secondary? Hydrogen bonding. What goes from secondary to tertiary? What are they? Hydrogen bond, excellent. Van der Waals and hydrophobic interactions, excellent. And to go from third to, we go from third to quaternary structure,
You add the
ionic bond on top. Very good, very good, very good.
on top of the other.
On top of the others, yeah. Okay. Whew. All righty.
Are you gonna ask like where the ionic interactions happen?
Oh, no. Just general. Transcription, translation. Okay, so use your study guide to look through these topics, and you can also online find your answers and use your